Yeah. Hard enough on a good day, you know, when you try to, are you screaming it?
Oh, yeah. You got to go out.
Hello. Hi. I'm Alice. This summer I conducted research in the Marco Lab, trying to understand if the Corsi gene influences bacterial sensitivity to two peptide bacteriocins, Plantarum and EF. So Lactobacillus plantarum was a bacteria that we worked with extensively in our research. It's a lactic acid producing bacteria, it's gram-positive. It's found in the human gut, so your gut, and it's also found in many fermented foods, so you've probably eaten it with kimchi or yogurt or cheese. It's been studied widely as a possible probiotic. However, what we were more interested in for our research was the fact that it can produce these small peptides called bacteriocins. So bacteriocins are these small antimicrobial peptides that bacteria can produce to kill other bacteria. Generally, the producer strain produces bacteriocins that target other bacteria that are genetically similar to the producer strain. So to simplify, if a bacteria is in a very highly competitive environment like the human gut, it makes sense that it would target other bacteria that need the same food as it so that it could survive. It's estimated that anywhere between 30 and 99% of all bacteria produce bacteriocins, which is why there's such a wide variety in their structure and how they kill bacteria. In this picture you can see that there's different ways that bacteriocins kill bacteria. So sometimes they just disrupt the cell membrane called cell leakage, or they attack certain receptors and prevent the function of those receptors, also causing cell death. So the specific bacteriocin that we were looking at this summer was Plantaricin EF. So Plantaricin is produced by Lactobacillus plantarum, and so L. plantarum actually produces about 20 different types of plantaricins, but we chose to work with EF specifically because of how much is already known about its function. We also knew that a sensitive strain was L. plantarum strain 965, and that the producer strain was L. plantarum strain 8826. Furthermore, we knew about its structure. So actually, Plantaricin EF is composed of two separate plantaricins, Plantaricin E and Plantaricin F. So separately, they aren't that toxic to bacteria. However, when they come together in equimolar ratios, they can form this helical structure, which they then use to bind to this protein on the surface of a cell. So this is the Corsi protein, and generally the way it works is Plantaricin EF goes, finds a cell that it wants to kill, and binds to this protein. And then through that protein, it disrupts the cell membrane and causes cell leakage and cell death. So the way that we know that it targets Corsi is because of some past research done in the lab, where that sensitive strain 965 was taken, its entire genome was sequenced, then it was exposed to Plantaricin EF until it became resistant, which you can see on this graph. So the green curve is the non-mutated 965, and then there's a blue curve, which is just growing regularly, which means that it's just resistant to the effect of Plantaricin EF. So when they resequenced the genome of this new mutated 965 strain, it was found that there was one nucleotide change that was significant in the Corsi gene. So the question we were trying to answer is, does the expression of Corsi induce sensitivity to Plantaricin EF? And I guess more specifically, it's that we were trying to see if certain differences in the Corsi gene correlate with more or less sensitivity to this plantaris EF, to this poison. So this is the way that we did it. Our data was actually comprised of two separate sets of data. The first was sequencing that Corsi gene, and the second was running bacteria through inhibition assays. Actually, so we used 3 different strains of L. plantarum, and then 9 different strains of L. pentosus. And so L. pentosus is just very closely related to L. plantarum, which was why it was chosen, since bacteriocins generally target bacteria that are genetically close to the producer strain. So in order to sequence Corsi, first we had to amplify that piece of DNA in all the bacteria's genomes, which we did so through PCR. This picture is a gel, which just proves that we amplified the Corsi gene, that we had something to sequence. So once we cleaned up all that DNA, it was sent off to be sequenced. And then we started on the second part of our data, which was running inhibition assays. So inhibition assays essentially just test the bacteria's reaction to plantaris EF. And so the way that it was set up is we ran them in 96 well plates. This is a map. And so all the strains of bacteria were grown in the presence of different concentrations of this plantaris EF. It ranged from 100 nanomolar to 0.78 nanomolar. Data was collected over 24 hours every 30 minutes, and the growth of the bacteria was measured through their optical density, through light. And this is what the results look like. So this is a super cleaned up version of an inhibition assay data. So this is just one graph of 12, since we have 12 strains. This is showing BGM48, which is an L. pentosus strain. And on the x-axis, you can see that it's time, because assays were run over 24 hours. And on the y-axis, you can see just a measure of how fast the bacteria were growing. So the way that we collected our data was, you can see on this blue curve, which is the control, this is how the bacteria BGM48 grows without any plantaris and without inhibition. And this red curve here is how it grows in the presence of the highest concentration of plantaris and EF that we tested. So you can see that on this blue curve, it took roughly about 6 hours for it to reach exponential phase of growth. And on this red curve, it took about 18 hours. Now that difference in time is called the lag period, and that's what our data was. That's how we collected our data. This is B1.3. This is a different strain. It's L. plantarum, and you can see here that it was completely uninhibited by plantaris EF, which shows that it's resistant to the action of this poison. BGM58 was the polar opposite. It was actually inhibited a lot by plantaris EF. As you can see, the lag period was greater than 24 hours. And this gray curve you see in the middle is just another concentration of plantaris EF that we tested. And it serves to show that as you increase the concentration of plantaris EF, the growth of the bacteria becomes more and more inhibited. This is a graph that has 3 strains on it, and it shows that different strains have different sensitivities to the plantaris EF. And so you can see this red curve completely uninhibited. The control and the tested bacteria are exactly the same, whereas this purple curve, although the growth of its control was pretty low too, it was inhibited a lot by plantaris EF as well. This is what the data looked like from the Corsi sequencing from the other half of our data. So it's just a tree that organizes the strains based on solely how similar they are based on their Corsi genes. This data was then turned into this tree, which shows how related the bacteria are based on the Corsi protein itself. So based on that protein that the plantaris EF binds to when it wants to kill a cell. This table here is all of the data collected from the inhibition assays. You can see, so each strain is listed and the lab time it had. In this red circle is where the strains were that were most inhibited by plantaris EF, and it suggests that since they're all in that location, they all have the same protein, and that if bacteria have a protein like that, then they're probably going to be inhibited or very sensitive to plantaris EF. So to summarize, the question was, do changes or differences in the Corsi gene lead to more or less sensitivity to plantaris EF? And the answer is yes, but also that more data needs to be collected. So the way that we could also prove that Corsi is involved in plantaris EF sensitivity is by taking the sensitive strain 965 and just taking out the Corsi gene. So then if that bacteria suddenly becomes completely resistant to plantaris EF, then that's proof that Corsi is indeed responsible for sensitivity. Another possibility is taking a previously resistant strain, such as Lactobacillus rhamnosus, and putting the Corsi gene in it. And again, if it becomes sensitive to plantar erythema, that's again proof that Corsi has a role in plantar erythema sensitivity. Just a further avenue of research is studying immunity proteins. So generally, bacteriocins can actually kill the bacteria that produce them, but the producer strains produce these immunity proteins, which block the action of their own bacteriocins against the cell. And so understanding how that works can just help us understand how plantaricins work more in general. In fact, so the research that was done here was pretty basic because it's an expanding field, but eventually the hope is that bacteriocins and plantaricins could be used in the food industry to understand how food could be better preserved or more pathogen-free. For example, there's already niacin, which is a bacteriocin that has been in use in the food industry since the 1950s, and it improves food preservation qualities. I would like to thank Dr. Mia Marco for letting me work in her lab, all of the amazing Marco Lab people, and especially graduate student Dustin Heaney for the incredible amount of time he spent with me working on this project. Thank you. So the question was, are there any other proteins that are similar to the Corsi protein? And so actually there was some research done in the Netherlands where it was found, so there's plantaricin EF and then there's also another plantaricin called plantaricin JK. So in the Netherlands they found that plantaricin JK tries to kill cells through a series of amino acid transporter. Yes, there are, but not exactly like that. Question was, can bacteriocins be used as antibiotics? And the hope is that yes. However, so antibiotics are kind of dying out because bacteria are becoming more resistant. And so more research needs to be done to see if bacteria could become resistant to bacteriocins as well. For example, like in our research, we actually made the 965 strain, which was sensitive to plantaricin mutate so that it was no longer sensitive. So yes, but more research needs to be done.
How long did it take for that strain to mutate to become resistant?
You know, I'm not sure because that was done before I got here, but I will refer you to the person who did it.
It only took about a week.
Which is pretty fast.
tyrosine sulfation in the Department of Neurobiology, Physiology, and Behavior under our mentor, Dr. Grace Rosenwitz. Tyrosine sulfation is the post-translational modification of the amino acid tyrosine, which is shown unsulfated right here. This process occurs in the trans-Golgi network and is characterized by the addition of a sulfate group from the universal sulfate donor PAX to the hydroxyl side chain of the tyrosine. And so this is where the sulfate group is added. This process is catalyzed by two enzymes, TPST1 and TPST2. Tyrosine sulfation occurs in a pretty specific environment. It always occurs in the extracellular domain, and then it's usually found in acidic and negative areas. So this is meaning that it's usually found near aspartic or glutamic acids. In addition, tyrosine sulfation is often found in tyrosine-rich environments, which leads to a phenomenon known as clustering, where at a certain predicted or known tyrosine sulfation site, there is usually another tyrosine sulfation site 5 amino acids before or after this site.
Tyrosine sulfation was first discovered by Frederick Bettelheim in 1954 in bovine fibrinopeptide B, a protein that assists with blood clotting. Later, in 1990, TPST, the enzyme responsible for catalyzing tyrosine sulfation, was purified from the bovine embryo medulla. In 1998, and the second version of TPST, TPST2, was discovered due to the high degree of homology shared with TPST1. Although the majority of tyrosine sulfation research has occurred with eukaryotes, tyrosine sulfation has long been thought not to exist in prokaryotes until 2012, where Pam Ronald demonstrated that it existed in the prokaryote Xanthomodus oryzae, PV oryzae.
So tyrosine sulfate can form up to 9 hydrogen bonds with surrounding amino acids and the enzyme substrates, so it can have a significant effect on protein structure and function. So tyrosine sulfate is especially important for protein-protein interactions and signaling. So it's found in proteins like G protein coupled receptors, blood coagulation factors, gastrin, and chemokine receptors. In fact, tyrosine sulfation is found in chemokine receptor 5, which must be tyrosine sulfated for HIV to infect white blood cells. Also, evidence of tyrosine sulfation is found throughout the human body, which also indicates its importance in biological function. So now I'd like to talk about my investigation into the PSM and the sulfonator. So the PSM and the sulfonator are two computational tools for predicting tyrosine sulfation in proteins. So we use computational tools to predict tyrosine sulfation because current detection methods like mass spectroscopy and labeling radioactive sulfur are expensive and time-consuming. So the PSM, or the position-specific scoring matrix, scores tyrosine sites, which consist of the central tyrosine and the 5 amino acids before and after it, using a table of scores for each amino acid at each position relative to the tyrosine. And then for each amino acid, the PSM adds up the scores for those amino acids, and then higher scores indicate higher likelihoods of being sulfated. We also, the PSM was also jackknifed, which means we removed one of the sites from the PSM and built a new PSM and scored the removed site with this new PSM. And this way, the effect of including a site in the PSM on the site score is removed. The second tool we looked at was the sulfonator. The sulfonator uses four hidden Markov models to predict tyrosine sulfation, and then it's available online, and it claims 98% accuracy. Unlike the PSM, the sulfonator has scores which lower scores are better and more likely to be sulfated. So here are the methods for my investigation. So we used a list of 102 P sites and 499 N sites, or 102 sulfated sites and 499 unsulfated sites. And we scored them using the PSM and the sulfonator. So from this list of 601 scores, we calculated the sensitivity, specificity, accuracy, and Matthews coefficient at different cutoffs for both the PSM and the sulfonator. And then finally, from these statistics, we derived a useful set of cutoffs for the PSM. So here's a graph of the correlation between the jackknife scores and the PSM. So the red scores represent the N sites, the unsulfated sites, and the blue diamonds represent the P sites and the sulfated sites. As you can see here, they're both very correlated fairly well with fairly high R-squared values. And then for the N sites, since we have 499 of them, the jackknife scores and the PSM scores are essentially the same. However, for the P sites, we only have 102 of them, and the jackknife scores are about 2 points lower than the PSM scores. So this illustrates that including a P site inside of the PSM boosts the score by about 2 points, which is why we use the jackknife scores to evaluate the PSM rather than just the regular PSM. We also looked at the correlation between the sulfonator and the PSM. As you can see by the spread of dots, or across the graph, the sulfonator and the PSM are not very strongly correlated, and thus they predict tyrosine sulfation very differently. As you can see, there are a lot of PSM sites with high PSM scores, but also high sulfonator scores, and then and the R-squared values are fairly low. So the main part of this investigation was looking at the sensitivity and specificity of PSM and the sulfonator at different cutoffs. So the sensitivity, or the true positive rate, is equal to the number of true positives over the total number of P sites. And the true positives are the number of positive sites correctly identified as positive, and then true negatives are number of negative sites identified as negative. So specificity is the number of true negatives over the total number of N sites. So sensitivity and specificity range from 0 to 1, where higher values are better. However, as you can see from this graph, as you increase the lower cutoff, you increase specificity at the sacrifice of sensitivity. So when you choose a lower cutoff for your computational tool, you have to balance sensitivity and specificity. For the jackknife PSSM, the sensitivity and specificity cross at about a cutoff of 0.5, and the sensitivity and specificity there is about 0.85. So in comparison, the sulfonator has a really high specificity across all of its cutoffs, but the sensitivity is fairly low. In fact, a sulfonator misidentifies about a third of the P sites at its default cutoff, which is why the sensitivity is really low. Next, we looked at the overall accuracy of this PSSM and sulfonator. So this green line represents the regular accuracy, which is the total number of sites identified correctly over the entire set of 601 sites. However, at the highest cutoff, where all the positive sites are identified as negative, the accuracy doesn't decrease very much because we have so many more N sites. So we also calculated the weighted accuracy, where the number of true positives and false negatives multiplied by the number of N sites, and the number of true negatives and false positives were multiplied by the number of P sites. So this way, the relative contribution of the P sites and the N sites are the same, and the two sides of the graph are roughly equal. So for the jackknife PSSM, the accuracy reaches about 90%, and the weighted accuracy reaches about 85%. If you compare it to the sulfonator reaches about an accuracy of about 87% and a weighted accuracy of about 76%. This is clearly not as good as the PSSM. So finally, we looked at the Matthews coefficient, which is a statistic for measuring how well a tool classifies data into two categories, in this case sulfated and unsulfated. The Matthews coefficient ranges from -1 to 1, where 1 means the tool's predictions are always correct, -1 is the predictions are always incorrect, and 0 is that tool's predictions are no better than random guessing. So here, the jackknife Matthews coefficient reaches about a maximum of about 0.65 at a range at cutoffs around 2.5 to 3. And while 0.65 might not seem very good, it's actually very good because most tools don't reach a Matthews coefficient of even of 0.7. So if you compare it to the sulfonator reaches a Matthews coefficient of about 0.55, which is not bad, but also significantly worse than the PSSM. So based on the data, we can see that the accuracy is much lower than 98% as claimed by the sulfonator paper. And this is somewhat of a problem since the sulfonator is used so widely to predict tyrosine sulfation in research. And then since the PSSM has better sensitivity, accuracy, and Matthews coefficient, the data suggests that the PSSM should be used as a primary tool to score tyrosine sites. However, the sulfonator is still good enough to use to cross-check PSSM scores that are low but positive. The data also suggests that the optimal cutoff for the PSSM is about around 2.5 to 3, since the accuracy and the Matthews coefficient are maximized at around that cutoff. However, we would also recommend cutoffs from 0 to 3, since depending on how important excluding false positives are. Hopefully this information will help researchers predict sulfation in new proteins and find new P sites which can help further improve our computational tools. I would like to thank Dr. Rosenquist for excellent mentorship and guidance throughout this project. I'd like to thank Ria and Rashida for their being excellent lab partners. I'd like to thank my counselor Anne for her support throughout the YSP program. I'd like to thank former YSP students Emily, Sabrina, and Jack for leading us through the initial steps of tyrosine sulfation and looking over our papers. Finally, I'd like to thank YSP and UC Davis for offering this wonderful opportunity. Next, my colleague Ria will talk about her research about PROs.
Good morning, everybody. My project here at YSP focused on predicting tyrosine sulfation sites and pattern recognition receptors. Pattern recognition receptors, or PRRs, are receptors that activate the innate immune response. Without these receptors, many organisms that lack adaptive immunity would not have even a basic line of defense against pathogens. Predicting sites of pattern recognition receptors can also help to develop drugs that target specifically innate immunity. Pattern recognition receptors can activate innate immunity by sensing two kinds of molecular patterns. These include pathogen-associated molecular patterns, or PAMs, or damage-associated molecular patterns, or DAMs. The detection of these molecular patterns then triggers downstream signaling cascades, which leads to the upregulation of genes that encode for molecules such as cytokines, which are essential to the inflammatory response. Although 5 types of PRRs exist, we will only be focusing on 2 of them, toll-like receptors, or TLRs, and selective type receptors, or CLRs, as these are transmembrane receptors and therefore have an extracellular domain. As Archita mentioned earlier, tyrosine sulfation only takes place in an extracellular environment, so we will only be focusing on these 2 types of PRRs. Additionally, multiple studies have shown that receptor kinases are essential to mediating plant innate immunity. And because these also have an extracellular domain as well, we will also be looking at these PRRs. These are just diagrams of the 3 types of PRRs that we examined. So as you can see over here, the TLRs, these have their exterior domain with the leucine-rich repeats, which is where the PAMs bind to. And this is the cytoplasmic domain that activates the downstream signaling pathways. And the CLRs, you can see these structures over here. These are carbohydrate recognition domains. So CLRs will recognize any sort of ligand that is a carbohydrate. So this includes mannose or glucose or glucans. And these are all produced by pathogens such as bacteria, fungi, or parasites. Finally, this is a diagram of 2 receptor kinases coming together to form a heterodimer in response to flagellin, which is a very common PAM produced by bacteria. Although tyrosine sulfation has been demonstrated to exist in the PAM AX21, which mediates innate immunity in rice plants, it is not known whether tyrosine sulfation occurs in PRRs, and hence that was the focus of my project. In order to generate a list of predicted positive sites in PRRs, I scored these PRRs with a PSSM calculator with 102 positive sites and 499 negative sites. I then used a cutoff of 1 and 2.42 to determine sites with intermediate scores and sites with high scores. A score of 1 was used as an arbitrary cutoff, as there are no known PRRs which are sulfated. A score of 2.2 was used for the high scores, as this number, determined through ROC analysis, minimized the number of false positive and false negative sites that were generated out of the calculator. Next, I then looked through PubMed to look for mutagenesis sites where the tyrosine site was mutated, as this would further substantiate the prediction that that tyrosine sulfation is necessary for the PRR to function. Finally, I used Muscle, a bioinformatics tool, and UniProt to align the sequences of the PRRs to look for conserved sites, as this would indicate some sort of an evolutionary significance. Out of my 84 PRRs that I scored, I generated around 130 P sites. Out of these 130 P sites, 37 of these had intermediate scores, so those would be scores in between 1 and 2.42. The average score for these sites was 1.65 plus or minus 0.42 standard deviation. Additionally, there were 38 sites with high scores, and so these scores would be above 2.42. The average score for these sites was 3.9 plus or minus 1.13 standard deviation. The rest of the sites were beneath 1, and so they were too low to be used for further analysis. This graph depicts a small snapshot of all the data that I gathered, and the green and red lines denote the cutoffs for intermediate and high scores. The green line over here is a set of 1, which is the cutoff for intermediate sites. So all the bars in between 1 and 2.42 represent sites with intermediate scores. The red line is a set at 2.42, which is the cutoff for high scores. So all of the bars above 2.42 represent sites with high scores. As our tutor mentioned earlier, clustering is also a common phenomenon of tyrosine sulfation, and so I looked for potential tyrosine sites that were located within 5 amino acids of each other and grouped them together in this graph. As you can see here, I found 7 clustering sites. 3 of these are in between TLRs. The yellow, the dark, the blue, light blue, and the dark blue are all CLRs, and I found one in one integrin beta protein. You can also see how the PSSM scores fluctuate between the clustering sites. For example, in the integrin beta protein, the scores are very similar. But in the CLR over here, you can see that the score is wildly fluctuating from a 4.2 to a 1.3. I also constructed a table to observe all the conserved sites in PRRs. This is just a small sample of the data that I gathered. So what's notable about this pair of TLRs is that not only is the tyrosine conserved, but the entire plus or minus 5 sequence is also conserved. And so these two TLRs have the same score. This is further evidence for strong evolutionary significance. Furthermore, on this phylogenetic tree, you can see that the TLRs 1 and 6, which also have the entire plus or minus 5 sequence conserved, are also next to each other on this evolutionary tree. This further indicates that these two TLRs have a strong evolutionary relationship. I then looked through literature to determine any mutagenesis sites found between the PRRs. These include both site-directed or induced mutagenesis and natural variants, such as single nucleotide polymorphisms. An example of a directed mutagenesis that I found was in the PRR racinosteroid-insensitive receptor kinase 1, found in Arabidopsis thaliana. At position 96, the tyrosine was mutated to an alanine, and it was found that there was a loss of heterodimerization, which then further suggests that the PRR's function was impaired. An example of a natural variant I found was in TLR4, where 3 amino acids upstream of the tyrosine and the aspartic acid was mutated to a glycine, and this mutation was correlated with an increased risk of macular degeneration or loss of vision in the eyes in Caucasian populations. Furthermore, when I rescored this new site, the PSSM score dropped from 2.85 to 0.99. So this new site with the mutation is less likely to be sulfated, further suggesting that tyrosine sulfation is vital for this PRR to function properly. Predicting sites of tyrosine sulfation in PRRs can further explain PRR ligand interactions. All of the sites that I predicted were located in the extracellular domains of the receptors, which is where ligand interactions occur. Thus, if PRRs are found to be tyrosine sulfated, and could potentially further explain the affinity of the ligand binding to the PRRs. As I mentioned earlier, PRRs can also be predicting tyrosine sulfation sites in PRRs can also lead to drugs that can target innate immunity. As found with the mutation in TLR4, which is linked with an increased risk of macular degeneration, if tyrosine sulfation is known to be a vital part of the PRR to function properly, further pharmaceutical research can assist in developing drugs that target that mutation. Finally, predicting sites in PRRs can elucidate the prevalence of tyrosine sulfation in the innate immune response. The binding of CAMPs to PRRs further regulates the transcription of cytokines. Chemokines, a type of cytokine, bind to receptors that are tyrosine sulfated, which is part of the entire immune response. Hence, if tyrosine sulfation is found in PRRs, it can explain the prevalence of tyrosine sulfation throughout the entire immune response, from the binding of CAMPs to PRRs to the upregulation of chemokines. As a final overview, took 3 types of pattern recognition receptors, TLRs, CLRs, and receptor kinases, scored them with a PSSM calculator, and generated a list of predicted pre-sites which were then further analyzed for clusters of mutagenesis and conserved sites. I would like to thank Dr. Rosenquist for her constant mentorship and guidance throughout this entire project, Arjita and Richard for being wonderful lab mates, Emily, Sabrina, and Jack, last year's YSP students, for guiding us through tyrosine sulfation and giving us feedback on our papers, and for her support throughout this project, and UC Davison's the Young Scholars Program for providing me with this wonderful opportunity. Next, my colleague Arjita will explain her research in tyrosine sulfation.
My name is Arshida, and I work on predicting tyrosine sulfation in the proteins essential to liver regeneration. The liver is the only organ in the human body that is capable of regenerating itself. And what's really important is as the liver regenerates lost mass, it is able to maintain the homeostasis and the processes like filtering toxins and regulating blood chemistry that are essential to survival. Hepatocytes are a parenchymal liver tissue cell that are the first cells to initiate liver regeneration. Most of the proteins that I looked at played a role in the initiation or expression of these cells. As the loss of parenchyma leads to liver failure, these cells play a very important role in the process of liver regeneration. All the proteins that I looked at, I scored in the PSSM calculator, which both Ria and Richard have mentioned. I used the general cutoff of 1, which was more of an arbitrary cutoff, and then I used a higher cutoff of 2.42, which was where the false positives and false negatives from the PSSM calculator were minimized. This cutoff was used to decide which of my sites were the most likely to be predicted. I next used the protein sequencing database UniProt to sequence my proteins and to also look for natural variants or site-directed mutagenesis. Lastly, I used the muscle alignment tool, an alignment tool to look at conserved sequences into my receptors. The first group of proteins I looked at were cytokines, and cytokines play a role in regulating transcription and the priming phase of liver regeneration. These are activation ligands that help to push hepatocytes from the G sub 0 stage of the cell cycle to the G sub 1 stage. In this group, I found one protein that was above my cutoff of 2.42. This was the tumor necrosis factor alpha, or TNF alpha, which at site 217 had a high score of 3.54. This protein plays an important role in the autocrine activation of the epidermal growth factor receptor, or EGFR. It also plays a role, as I mentioned earlier, in the priming phase of regeneration, so this includes signaling and tissue formation. The next group of proteins I looked at were the growth factors. These proteins play a role in the progression phase of liver regeneration and help to push hepatocytes past the G sub 1 restriction point in the cell cycle. In this group, I found 11 sites with an average of 4.61 and a standard deviation of plus or minus 1.54. This is just an overview of some of the highest scores I got in my growth factors group. As you can see, the highest score is 8.77, which I found on the transforming growth factor, beta 2, at site 57. This protein plays an important role, along with activin, another protein in liver regeneration, in terminating hepatocyte proliferation. This means that this protein actually plays a role in ending the process of liver regeneration. The third group of proteins I looked at are cytokine and growth factor family receptors. I found 21 predicted sites in these groups with PSSM scores ranging from 2.42 to 5.78. I had an average of 3.28 and a standard deviation of plus or minus 0.89. What was really important about some of the receptors I looked at in this group was that I found natural variants at 4 sites. All 4 sites were in fibroblast growth factor receptors, or FGFRs, and they were in FGFR1 and FGFR2. Both of these receptors are in the fibroblast growth factor family, and they're tyrosine kinase receptors. Over here, I have a chart of the different subfamilies that these receptors bind to. So there's 22 fibroblast growth factor members. However, these members are divided into 7 subfamilies. And so as you can see, these 2 receptor proteins bind to different subfamilies. For example, fibroblast growth factor receptor 1 binds to FGF2 in the subfamily FGF1. However, FGFR2 does not bind to this growth factor. Similarly, an FGFR2 binds the fibroblast growth factor 7 subfamily, which FGFR1 does not bind to. Next, I did an alignment of these two receptors because I wanted to see how much of the sequences between these two proteins were conserved. As you can see, the shaded regions are where the sequences are completely conserved, which means the amino acids in these regions were not changed at all. I have a zoomed in section of the boxed out section from below, where the FGFR1 sequence is on the top and the FGFR2 sequence is on the bottom. The box that's highlighting two of the Y's is where the tyrosine sites are that I predicted natural variants. And what's really important is that this sequence between the two proteins is completely conserved, which means that the PSSM scores from this tyrosine site are going to be completely the same, as you will see in the next couple of slides. So the first site was in fibroblast growth factor receptor 1 at site 339, which had a score of 0.69. When the tyrosine was changed to cysteine, individuals were shown to contract Kalman syndrome, which has a wide spectrum of phenotypes, but is characterized by the failure to start or finish puberty. It's also associated with a lack or reduced sense of smell. In fibroblast growth factor receptor 2 at site 340, which had the same score of 0.69 because, as I mentioned earlier, these sequences are completely conserved between the two receptors. When the tyrosine is changed to cysteine, individuals were shown to have Pfeiffer syndrome, which is a craniosynostosis syndrome, so that means that there is a premature fusion of skull bones in these individuals. However, there's 3 types in Pfeiffer syndrome, with types 2 and 3 being more severe and often associated with nerval issues. At the same site in FGFR2, when tyrosine is changed to histidine, individuals were shown to have Krausen syndrome, which is still in the craniosynostosis family. However, this syndrome is associated with hearing and dental issues. The last natural variant I found was still in FGFR2, however, it was at site 375 with a score of 2.03. While this didn't quite make my high score cutoff of 2.42, this is still a relatively high score. When the tyrosine was changed to cysteine at this site, individuals were shown to have Behr-Stevenson cutis gyrata syndrome, which is still in the craniosynostosis syndrome family. However, the cutis gyrata portion of the name comes from the skin abnormality that is associated with this syndrome, which is basically the furrowing of skin. And lastly, I looked at an array of additional proteins that helped in supporting the other groups of proteins that I looked at or played their own individual role in the process of liver regeneration. Most of them were ligands that bind to some of the receptors that I talked about earlier, and they play a role in the signaling pathways and protein-protein interactions in the process of liver regeneration. In this group, I found 13 sites with an average of 4.44 and a standard deviation of plus or minus 2.72. This is a chart of one of the most important proteins I found in the additional protein group, which was amphiregulin, which had the highest scores of actually any of the proteins I scored, and those scores were 7.78, 10.92, and 9.03. What's also really important to notice here is that these sites were clustered, so there are 81 and 83 and 87. And as we mentioned earlier, when clustering shows a higher probability of tyrosine sulfation. Amphiregulin is a ligand of the epidermal growth factor receptor, or EGFR, and like TNF-alpha, which I spoke about earlier, it also plays a role in the autocrine activation of EGFR. This is just a final overview of the proteins I looked at that played a role in the process of liver regeneration. I looked at initiation, which is caused by cytokines, and then progression, which comes from the growth factors. I next looked at receptors, which play a role of binding, and so they support both cytokines and growth factors. And then I also looked at an array of additional proteins. Most importantly, the one that I highlighted here was amphiregulin, which helps to support the process of liver regeneration and the other 3 groups of proteins I looked at. What's really important is that I predicted tyrosine sulfation in all 4 of these protein groups, which means that potentially tyrosine sulfation plays a very substantial role in the process of liver regeneration. However, further research is needed to experimentally prove the existence of the tyrosine sulfation sites because the information that I have gathered is just prediction of tyrosine sulfation sites. However, the information that I have gathered will help to will help with the general knowledge of tyrosine sulfation and could potentially play a role in aiding medical researchers in finding treatments for those with weak or impaired liver regeneration. I'd like to thank my mentor, Dr. Rosenquist, for her amazing support and help throughout this project. I'd also like to thank my lab mates, Richard and Bria, who supported me and helped me in my collection of data throughout this project. I'd like to thank last year's tyrosine sulfation group, Emily, Jack, and Sabrina, for their help in introducing us to tyrosine sulfation and then reading over our papers. I'd like to thank my counselor, Ann Ha, and the Young Scholars Program for providing me with this opportunity. Thank you. Does anybody have any questions?
Why do you use the cutoffs 1 and 2? So for our cheetah and with our projects, we use the cutoff of 1 as sort of like an arbitrary cutoff because, at least in my case, there weren't any known pattern recognition receptors that were proven to be sulfated. And so the score, besides the scores in between 1.2 and 2 were determined to be cited with intermediate scores. For the number 2.42, this was determined in a paper long ago through ROC analysis that the specific score as a cutoff minimized the number of false positives and false negatives that came out of the PSSM calculator. So we also used that cutoff to determine seismic ice scores.
What makes sulfation so important?
So the question was, what makes sulfation so important? So common examples of sulfation, it really helps with protein-protein interactions and cell signaling. So an example of this was in chemokine receptors, there's a certain portion that is required to be sulfated in order for HIV to bind to it as a co-receptor. So tyrosine sulfation is also found in coagulation factors in blood and T protein coupled receptors as well. So it's important for self-constitution in general.
Alice, you actually mentioned autocrine activation for your presentation.
So the question was, what was the autocrine activation that I mentioned in my presentation? Autocrine activation is basically when a cell releases an agent that activates a receptor on itself, and then this causes a change within the cell. So it's when the cell basically changes itself.
Okay, so for the question was, how is the table of values for PSM generated? So the PSM is generated through an equation called Hennicoff's equation, which basically looks at the relative frequency of amino acids in P sites and relative frequency of amino acids in N sites. And then since we only have 499 N sites and 102 P sites, these are also modified by pseudo counts, and then they're based on the mutation frequencies of amino acids into other amino acids, and then the P sites and the N sites are basically the frequency of the P sites and the frequency of the N sites are divided, and the log of that is taken. So then to generate the and then this is done for every amino acid at each position.
Is the PSSM only used to examine tyrosine sulfation, or are there other uses of it?
Okay, so yeah, there are the PSM can be used to predict things and predict a lot of things, and then you have to generate PSM based on known data for each situation that you're using it for.
What exactly was the jackknife value that you mentioned in your slide?
Okay, so essentially, the problem, since we don't have a lot of P sites and N sites, we have to evaluate the PSSM. We don't want to, the scores might be boosted because the sites themselves are in the PSSM, right? So for the jackknife, we removed each site, and then we built a PSSM with all of the other sites, and then we scored the removed site with the PSSM made from the other sites. So, and then this is the jackknife scores. So then this way you can score the sites without including the effect of including itself inside the PSSM.
What does the high score from the PSSM mean?
So high scores from the PSM basically they correlate with like how likely is the site to be sulfated. So scores like above 2 were determined as sites with high scores. So these scores had an extremely high chance. These sites had an extremely high chance of like having a sulfate group attached to it. So that was the whole basis, is that we were trying to predict whether each site, what is the probability that the site is sulfated.
Is there like a number for a 100% chance of tyrosine sulfation on a site?
Well, so far of the N sites we know, the highest score known is like somewhere around 7 or 8. So anything above 7 or 8 is pretty much 100% chance to be sulfated. Although it is possible we might discover new N sites with higher scores. And then similarly, the lowest known P site score is about 0.7. So anything below that is almost certainly going to be unsulfated.
This is for Akita. So you talked about like a variety of syndromes with like a variety of symptoms and phenotypes. But like those syndromes, and you also talked about liver regeneration. Are those syndromes like directly connected to the liver regeneration, or are they just on 2 different spectrums?
So the symptoms that I looked at were, they're 2 different things. They're not directly connected to liver regeneration, but it was the fact that the tyrosine, when it's changed to another amino acid, cause a syndrome, because when an amino acid is changed, it usually doesn't cause this like a syndrome. But if the tyrosine is sulfated, then it could potentially cause a syndrome. So what's important is that the causing of syndrome implies that it's more likely for these sites to be tyrosine sulfated, which then will play a role in the process of liver regeneration.
What makes tyrosine like the prime recipient of that sulfate group compared to like other amino acids?
So tyrosine, multiple amino acids can be sulfated, but it's just like in this particular phenomenon, the tyrosine sulfation is just for this specific amino acid for tyrosine, because it's like the transferring of the negative sulfate to a polar amino acid, it increases the number of hydrogen bonds that can be made. And so in turn that really helps, that's why it helps with protein-protein interactions and cell signaling.
It's lunch.
Now we're going to have our lunch break.
Thank you. Archita, if you could come up here, there's somebody in India that would like to say hello. There you go.
Thank you.
You're welcome.